In the discussion of acidification of urine, mainly we need to say how the regulation of acid base balance is there, right? So the regulation of acid base balance is a very important thing in your acidification of urine. So what can be the two ways by your kidneys which can make your urine acidic? So basically the pH of urine is 4.5 to 6. Okay, 4.5 to 6. For making this urine to be acidic, your kidneys have to do either two ways. What are all those two ways? One, either they have to reabsorb the bicarbonates, right? They can reabsorb the bicarbonate ions, or else they can remove by the secretion or the excretion process of hydrogen ions. So any of the one thing can happen to make the urine acidic. But how it is happening, that is important. If at all we are speaking about bicarbonate reabsorption, it can be made by very important ways. Okay, so it can be filtered by glomeruli every day, right? It can be filtered, it can be excreted in the form of urine, it can be taken back, anything can happen. But the ways which are taken for acid base balance, so bicarbonates must be taken back from renal tubules. Remember that it is taken back from renal tubules, right? So how can the secretion of hydrogen ions can happen? Either the reabsorption of bicarbonates make the secretion of H plus. This can happen in the passive way, or else the other mechanisms for hydrogen ion secretion, which can be secreted in your renal tubules by carbonic acid way. Okay, so what is the other way that can be secreted? Like your H plus ions can be secreted, like in your carbonic acid, when they immediately dissociates, they'll give H plus plus bicarbonate. So by that way it will give H plus, it will secrete H plus. So it is secreted in the lumen of our PCT, in our PCT, in our DCT, and in our collecting duct. In all the places it is secreted, especially by help of which cells? Please remember, eye cells. Okay, here we come the importance of eye cells for handling your bicarbonate and hydrogen ions. So the secretion can be done in two ways. Okay, so what are all those two ways for secretion of H plus ions? One is your sodium hydrogen pump. Okay, one is your sodium hydrogen pump, and the another one is your ATP driven proton pump. Because of this ATP, your proton pump is working. So this is how the ways your urine can be acidified, either the reabsorption of bicarbonates or the secretion or removal of hydrogen ions. So what they are happening exactly? What if they are happening exactly? When there is a sodium and hydrogen pump, what is happening in this pump? So whenever there is a sodium reabsorption from your fluid, okay, your H plus is secreted, which is what we have seen in our, yes, in our proximal convoluted tubule, right? There will be an exchange. Because of this antipode system, because of this antipode exchange, we can say, right? In this antipode exchange, which is responsible for our sodium and hydrogen exchange, by that way hydrogen is secreted, right? This occurs in our PCT also, but mainly we can expect this in our DCT, but most probably reabsorption occurs maximum in your PCT, please remember this. But here sodium hydrogen pump antipode exchange, where secretion of H plus ions are occurring, that is happening in your DCT. What is the second thing which we have seen? That is your ATP driven proton pump. ATP driven proton pump. What is happening in this ATP driven proton pump? The name itself says that there is responsibility of ATP, right? H plus is secreted in your DCT and in connecting duct by the help of ATP. H plus secreted, that's it. So these are the two ways how your H plus can be secreted and removed. So now in your acidified urine, your H plus ions should be excreted. Okay. Now we have seen how it is secreted. So, but the excretion of H plus ions which makes your urine acidic, urine pH acidic. This excretion of H plus ions, further it is having three other mechanisms to be remembered. Either by your bicarbonate mechanism it can happen, okay, or by your phosphate mechanism it can happen, or by your ammonia mechanism it can happen. So three other mechanism for removal or excretion of H plus ions to be remembered. So in this picture we are going to see how the reabsorption of bicarbonate happens, which makes our excretion of hydrogen ions. See, in our lumen there is sodium bicarbonate, which is a filtered urine now, the concentrated thing. This getting separated between sodium and bicarbonates. Once they are divided into sodium and bicarbonate, what they'll do, from the lumen to our tubular cell gets transported. Our sodium gets transported. Further, our bicarbonate ions, they will be acting along with our H plus. How? These H plus are utilized because of, please remember, because once the sodium goes from your lumen to the cell, the counter current secretion of H plus happens. Okay, this is happening because of your sodium goes in and the hydrogen ions comes out because of your bicarbonate absorption. So this bicarbonate absorption always takes place in your PCT. Remember, we know that maximum absorption takes place in PCT, and if you speak about bicarbonate absorption, that also takes place in our PCT as well. So once this is secreted, H plus is utilized. Here there will be a responsibility of a enzyme that is important for your carbonic acid. This is your H2CO3, which is formed after correlating with your hydrogen ions. Here there is an action of enzyme which is called as carbonic anhydrase. Once this enzyme gets activated, the formed carbonic acid gets divided into carbon dioxide and water. So this will be going into our cell. Okay, our cell needs water and carbon dioxide. Further, this gets dissociates in our cell. This is a cycle of dissociation and association. You can able to see here. They are getting formed and they are getting dissociated, but the only thing which comes inside again and again in your circulation, that is your H plus, please remember. So that is how this excretion of H plus also happens by your bicarbonates. Here which pump is used? The pump which is used here is your sodium hydrogen pump, and that too we can say it is in opposite direction, so it is a antipode pump. Clear? So the main enzyme which is used, how it is transported here, which is the important filtrate factor which is needed for our body, HCO3. So because of this reabsorption, our H plus gets secreted, right? In this picture, the second mechanism which we have seen is our reabsorption of, yes, what mechanism? Our sodium hypophosphate mechanism we are going to see. Because of this phosphate mechanisms, how your excretion of hydrogen ions takes place. Okay, so by this way, actually this is a mechanism, not a reabsorption. It is a phosphate mechanism by which your excretion of hydrogen ions takes place. If you can able to see in your tubular cell, always you know there will be a formation of carbonic acid by water and carbon dioxide collection. Because of this reaction, again your carbonic acid dissociates with the help of a carbonic anhydrase enzyme into bicarbonate and H plus. Further, this H plus gets reacted with our sodium hydrogen phosphate. Clear? Sodium hydrogen phosphate. So once this H plus have been added with our sodium hydrogen phosphates, they converted to form sodium dihydrogen phosphate. Okay, so what is the importance of the sodium dihydrogen phosphate? Why they are getting means formed with our sodium hydrogen phosphate? Because then only they will be sufficient to remove our H plus. So removing our H plus is a main target, right? So pH should be acidic. In whatever ways they can remove, they should remove that. By that way, now our hydrogen ions combined with our sodium hydrogen phosphate to form sodium dihydrogen phosphate. By that way, it is secreted. So this is very important to understand how this formation of H plus happens in your tubular cell, which goes on to your tubular lumen, and then it gets react with your sodium hydrogen phosphate to form sodium dihydrogen phosphate. It happens mainly in your distal convoluted tubule and collecting duct. Why? Because if you can able to see in this step, see sodium is there. How sodium is reabsorbed? Sodium is reabsorbed only with the help of a role of aldosterone, right? Without aldosterone, sodium will not be absorbed. Until and unless if we speak about aldosterone, that is a whole part in your distal convoluted tubule and collecting duct. Very important. The one more mechanism which is left is our ammonia mechanism. But how excretion of H plus happens? So we have seen many mechanisms by removing of excretion of H plus, but this is the most important mechanism for removing your H plus ions. So most important mechanism. And why your urine is acidic, the first importance should be given for this ammonia mechanism. Please remember, okay? Ammonia mechanism. So what happens over here? What you will understand? See, in your tubular epithelial cell, what is there? The ammonia which is formed by amino acid as glutamine. Amine, it is there. So this is formed from amino acid. We all know that, right? How glutamine is formed? With the help of the enzyme glutaminase, right? You could have studied in your biochemistry. By that, if it is formed, this converts our glutamine to glutamic acid, right? How? In this conversion, only your ammonia is also formed. This is called as deamination of amino acids, such as some other amino acids like glycine, alanine. They also produce ammonia, right? So we have seen a simple amino acid levels how they produce ammonia. So once the ammonia is formed in your cell, they will form only in your cell. So once this is excreted, what they will do? They'll go to your lumen. From the cell, they will go to lumen for exchange. Okay, instead of what? Instead of sodium, our ammonia will go there. Okay, once our ammonia will be going there, the tubular cell membrane is not permeable to ammonium. Please remember, first thing, our tubular cell, like other mechanisms, it is not permeable to ammonia. Okay, so what they will do? Once they are not permeable to ammonia, they'll change the structure. Okay, so ammonia which is formed in your tubular cells into the tubular lumen, they'll go for exchange. Here when it is combining with NH3, right? NH3, when it is combining with our H plus, right? Our H plus, here you can see. So this NH4 is not permeable to ammonia. It is not permeable to our lumen. Again, it cannot go back. So once our NH3 is permeable, same as like exchange with our sodium, but when it is secreting along with our H plus, this NH4 is not permeable. But our NH3 is permeable, remember. Once it is not permeable, they have been excreted in our urine. How? along with our H plus, when it is getting converted, they get excreted. Very, very simple. So rest of the things what they'll make, obviously in cells, are water and carbon dioxide get converted to form carbonic acid, then our bicarbonates will be there and hydrogen ions will be secreted. So this bicarbonate will be joining with the sodium which is coming from lumen. Sodium which is coming from lumen, it's get attached and they form sodium bicarbonate and they'll circulate in the fluid. So this is what the whole mechanism of 3 steps of your phosphate, bicarbonate and ammonia mechanism says. So here also we have some clinical applied part over this. So we have seen how the concentration of urine is, how the regulation of acid-base balance, what happens to secrete H+, what happens for absorption of sodium, absorption of bicarbonate, every process, right? So regarding that, if at all we are speaking about a clinical part, what happens if there is any abnormality in your functions, right? That is what we are going to see. That please remember, always in your PCT, which is the pump which is carried out, ammonia mechanism. Please remember, because of ammonia mechanism, H plus is secreted. You know that H plus is secreted in PCT also, but how? Because of ammonia mechanism. And in your DCT and collecting duct, who is taking place? Yes, your ATP driven pump, which we have seen, right? And also your phosphate mechanism. And along with your PCT, DCT, loop of Henle. In these 3, the main thing making over is your bicarbonate mechanism. Remember this. So apart from this, only in DCT, that is your sodium hydrogen pump. Okay. So if at all they ask only DCT, then your answer should be sodium hydrogen pump. If they ask in only PCT, then now it is ammonia mechanism. If they ask it bicarbonate mechanism, it happens in 3. But in collecting duct and DCT, it happens only by ATP and phosphate. Very, very important to remember. In the clinical part, if at all we are seeing about metabolic acidosis, when our whole metabolism gets acidosed. So what will be the reason? For example, your kidneys are failing to excrete your H+. That is what happens, right? Your kidney is failing to excrete H+, then your whole body will be secreted along with H+. So if they are failing to excrete the hydrogen ions, then they will lead with metabolism of acidosis. Okay, metabolic acidosis. Then what happens in metabolic alkalosis? Kidneys are excreting too much of excrete quantity of hydrogen ions. So too much of large quantity of H+ is excreted. So that is why you have alkalosis. Alkalosis means your body's alkaline condition is more because of too much of excretion of hydrogen ions. This both happens whenever there is a impairment or pathological problem in your kidneys, which we would be discussing detailedly in our medicine vast subjects. But up to now, in a physiological point of view, if you know about this clinical part, that is well and good. So now we'll be going to discuss about some parts of our regulation part. Now we have seen regulation of acid, right? Regulation of base and regulation of balances and what are all happening within them. So till now we are going to see some regulatory parts of our H2O and electrolytes. What happens within our regulation of our H2O and some of our electrolytes. So what you have to understand by this point, if at all there is regulation of water by kidneys, what you have to keep it in your mind. Either they'll be very normally by transcellular route or by paracellular route, right? You know that what is transcellular and paracellular, which we have discussed already, right? Either by transcellular route or by paracellular route, which we know already. So, and also we know there is a two type of H2O absorption, that is your facultative absorption and yes, obligatory absorption. So, right. So what happens in your obligatory absorption and what happens in your facultatory absorption? What happens in your facultative absorption? We are speaking in terms of water, okay, in terms of H2O. So what is happening within them? Obligatory means H2O absorption takes place from your PCT, which is very important. Normally H2O absorption takes place. PCT, the absorption is taking place. Okay, right. So what happens in facultative? H2O absorption takes place from your DCT and collecting duct. That is what your difference in obligatory reabsorption and facultative reabsorption. Then the next step is that ADH necessary for PCT absorption? Already I said you this as a question. Antidiuretic hormone is not necessary, okay, not necessary for your obligatory absorption. But in facultative, if at all they are happening in DCT and collecting duct, for sure your antidiuretic hormone is needed, it is necessary. Then what else left? We have read some channels, right? Those channels are also very important for obligatory absorption. So what are all those channels? In your obligatory, they are AQ, aquaporin 3 channels, right? In your DCT or in your facultative mechanism, the aquaporin 2 channels are involved, okay? So if I'm speaking about the obligatory absorption, do they have any issues or any influence in tonicity of the urine or tonicity of the fluid? Because as such urine is not formed in our proximal convoluted tubule absorption. So this H2O absorption in your PCT will not influence the tonicity of fluid, will not influence the tonicity, please remember, okay? So what about your facultative? It will influence, right? It will increase the tonicity, it will increase the tonicity. So these are the very few differences which you have to know at least in point of regulation of H2O. Please remember ADH is not important for your obligatory mechanism, right? So these are all the very important things, and you should know what are all the things, which factors that influence your H2O absorption. Who can influence H2O? Which we have seen, right? Who can influence H2O? Your GFR influence H2O, your aldosterones influence H2O by the way of sodium, right, in a passive way. Your ADH can influence H2O, and sometimes your angiotensin 2 can influence H2O, right? And then what happens? Your diuretics can influence also H2O. Diuretics can also influence H2O. So this is what happens in your influence of H2O, if at all we are speaking about regulation of H2O. Okay, so that is very, very important, which you have to understand in the point of view. Now if we are speaking about some physiological changes are also have been admitted. Physiological changes in intake of water. In a daily life, you cannot take the intake of water in a very normal way. Sometimes it can differ, sometimes it can increase, sometimes it can decrease, right? So how do your renal system gets managing that if there is a water deprivation or if there is increased intake of water? For example, the first case, if we see, there is water deprivation. You are taking very less amount of water in your daily life so that your body cannot able to equalize the fluid in content of your body. So what happens in your water deprivation? The plasma's osmolality increases. So what happens there? Increase in plasma's osmolality. Okay, increase in plasma osmolality. Once the plasma osmolality is increased, what they'll do, they'll stimulate your hypothalamus, right? So whatever things happens in your body, they will stimulate your hypothalamus. By which receptors? By osmoreceptors. There is a stimulation of hypothalamus. So once it is done, this hypothalamus can do either of the two things. What they can do, either they can stimulate thirst, okay, either they can stimulate thirst, or else they can do ADH release. ADH increase. Why they are increasing ADH? Because they are absorbing your H2O. Simple. But ADH is released from which gland? Please remember, posterior pituitary gland. Please remember, posterior pituitary gland. Simple. Now, so once these both things are done, once in our oral stimulation, what can happen? Once the oral stimulation, matlab in thirst stimulation, there will be increased oral intake, then you will drink more water because your thirst stimulation is indicated, then increased oral intake will be there. Further, your blood volume and osmolality will be normalized. So, blood volume and osmolality of the blood is restored, right? Whichever become decreased and whichever altered will be restored. Then here, because of this ADH, what happens? Because of this ADH, what happens? Water absorption from PCT or DCT? Water absorption is from obviously DCT. Once, if at all you are seeing ADH, they will be DCT in collecting duct. Then your urinary volume decreases. Urinary volume decreases because there is no water for excretion of urine. Everything is getting absorbed and your urine osmolality increases. Okay. By this way, your body restores your thrive or whatever the requirement. So this happens in the way of water depletion. So if at all I am speaking about, this happens in your water depletion. Okay. First one. If I am speaking about the second case, what can happen in the second case? There will be more intake. You are drinking too much of water. So there is increased water intake. So what can happen? Either two of the things can happen. First, plasma's osmolarity will be decreased. These both are always vice versa. Okay. So what happens? Decrease in plasma's osmolality. So once this is decreased, again the same thing happens. But your hypothalamus now here is inhibited because you are already having too much of water. This minus here denotes inhibition, inhibited. So once this is inhibited, again what happens? ADH secretion increases or decreases? Yes, ADH secretion decreases. So because of this decreased secretion of ADH, absorption will be decreased, right? So the absorption of H2O is decreased. Absorption of H2O is decreased. Why? Because ADH secretion is decreased or inhibited also, you can say. So once this is decreased, then what is the most probability? Obviously urine output will be increased because there is all the water is getting excreted. They are not getting absorbed back into your body. So urine output is increased. Once it is increased, so obviously it is dilute in nature. So the too much of water which is present in your body, which is now excreted as dilute urine. Now because of this, your blood volume decreases and it's restored. Blood volume decreases and osmolality is restored. So this is how the altered osmolality is getting restored by the way how our hypothalamus and the stimulation process are maintained. So this is the two things in which there is a first water deprivation we have discussed, and second we have discussed about the increased water intake. These both are a kind of a physiological changes in your body.